Alright, so this video is covering lecture 3, where we're going to be going over the first 3 macromolecules, carbs, lipids, and nucleic acids. We're also going to touch back on hydrophilic, hydrophobic one more time, and the criteria for a hydrogen bond. If you want to look ahead, this is kind of where we're going into. Carbs, we'll go over a lot of structural stuff, and that's it. So let's get to it. I'm going to try to keep this video under 10 minutes because your time is precious. Alright, so where'd my pen go? So hydrophilic, hydrophobic. Just know that hydrophilic is water loving, hydrophobic is water fearing. Let me get my pen set up. Also know hydrophilic is polar, is soluble, is hydrophobic is nonpolar. Insoluble. This is very important to know. This isn't the first time you guys are going to see this. When you guys are in organic chemistry, you're actually going to use the hydrophobic effect, more so polar and nonpolar, to make these things called aqueous and organic layers, because some things can dissolve in the organic, which is more desirable because it has a lower evaporation temperature, and then you can extract these things out of the polar and nonpolar layers. It's called immiscible liquids if you want to look up like extraction techniques on that, or just wait till you get to OChem. So when we're looking for hydrogen bonds, let's zoom in here. You're going to want to look for an NH, OH, FH. And these are capable of making the hydrogen bonds. So here we have an OH. This H could bond to something, and that would be a hydrogen bond. This can't be a hydrogen bond because there's no H hooked to this. Remember, we're looking for NH, OH, FH. If it doesn't have that, it's not going to hydrogen bond. This won't hydrogen bond, but this will hydrogen bond. This H could hydrogen bond. Down here, don't let this trick you because it's a nitrogen. Remember, nitrogen, oxygen, fluorine, all of those, if it's hooked to hydrogen, can hydrogen bond. That could hydrogen bond, that could hydrogen bond. There's even a lone pair of electrons, which is less likely, but those could as well. Down on this one, that could hydrogen bond, that could hydrogen bond. Alright, so when we get into carbohydrates, just know carbohydrates are linked together. The monomers are linked together by glycosidic bonds. You're going to want to know that these are extremely polar if you look at all these OH groups. So obviously these things are going to be pretty soluble in water. Whoa. And also know these structures. Memorize what this looks like. Trust me, you'll want to memorize what these structures look like. We have a linear structure, and then we have cyclic structures. These aren't the same, but they fold up. So basically this folds up into there, and it creates this structure. Know what these look like. Know that these can link up and form these glycosidic bonds, and it can turn into these really complex looking structures. Don't get this mixed up with a steroid. A steroid does not have all these OHs. You also might see it drawn like this. This is called a chair formation, and I keep referring to organic chemistry, but when you guys are in organic chemistry, you're going to see this chair formation. Chair formation. You'll remember me saying this when you get to OChem and you have to draw hundreds of these. So all 3 of these variants is what you might see when you're asked something structurally about carbohydrates. Alright, so when we get to lipids, I picked this slide to talk about lipids because it shows a triacylglycerol. And lipids, their form tells you a lot about their function. So here we have basically this thing, and it's connected to 3 long lipids. So this is a triacylglycerol, It's very good for energy storage. The main thing you're going to see this with is energy storage, because these can break off and be used for whatever, to make acetyl-CoA or something like that. So also understand that all of this whole part of the lipid chain is hydrophobic. This part is hydrophilic. So let's say there's a molecule, there's a protein inside the fluid of a high concentration of lipids. So let's draw it up here. Here's lipid heads. Lipid heads, fatty acid tails. Alright, you might see something like this. I know you saw it in your practice exam. So we have this molecule. We'll use this cool color again. It's basically just chilling in here. Get these other, there's more lipids down here. Boom. So this molecule is just chilling in here. And you might be asked, what proteins are expected to be on the exterior of this thing, this globular protein that's in here? Okay, so this requires a couple steps. So don't freak out and think that you don't know it, because you just got to break it down. So we know that this is hydrophobic, and all of these here are hydrophobic. So we know this area is very hydrophobic. Therefore, most of the liquid in this area is hydrophobic. Therefore, the exterior of this protein is going to be hydrophobic. Remember that hydrophobic attracts hydrophobic, hydrophilic attracts hydrophilic. They want to be with each other. This isn't like charges where opposites attract. Okay, so when we get to lipids and the actual cell membrane, you have saturated fats and unsaturated. So I'm just going to put sat and unsat. So saturated fats will look something like this. Okay. Cool. So, I shouldn't have done that. Okay, here we go. Okay, I drew this super janky, but it'll explain it. So saturated fats don't have any double bonds, and they're compacted very tightly, whereas unsaturated has double bonds, and this creates kind of these big open spaces. So when we look over here, why does that even matter? Why am I even telling this to you guys? Well, a cell that has a membrane that has more saturated fats in it is going to have a much more stronger membrane. And that's because these van der Waals forces are going to look more like this, where an unsaturated would look more like this. So just take that into account. Something that has more unsaturated fats in its membrane is going to be a lot more permeable. And just look at these big gaps, like, I don't know, maybe like picture you're a running back or something. I don't know, maybe you snowboard and these are just like huge lines that you can take, whereas this one, it's a lot more tight. This would be like going through the trees or something. Hopefully that analogy helps explain it. So when you look at nucleotides, memorize what these are composed of. We have a phosphorus group, we have a sugar, and we have a base. P, sugar, base pair. Know this. Know how these connect. Know how they will add on to each other. If there's a OH right here, if you see where my clicker is, this OH tells you that this is ribonucleic acid. This one doesn't have this OH, so it's deoxy. Deoxy means that it's less oxy. It has one less deoxy group or whatever right there. That little alcohol group is not there on this one, and that's how you know it's deoxyribose. So definitely understand how phosphate hooks to the sugar, which hooks to a base pair. These base pairs will hydrogen bond between each other. So then we'd have another base pair, sugar, boom. And this would just keep repeating up, and you'd have your long DNA strand going up and up and up. Alright, so we just hit 10 minutes, so I'm going to call this video, and I'll get started on lecture 3.